Welcome to today's webinar. My name is Lauren Perry. I'm Operations Manager at SRF and a parent to a 12-year-old with Syngap 1. Our presentation today is Harnessing Messenger RNA Metabolism for the Development of Precision Gene Therapy. And I have the pleasure to introduce today's speaker, Dr. Jeff Collar. Dr. Collar is a Bloomberg Distinguished Professor of RNA Biology and Therapeutics at Johns Hopkins University. His lab has made seminal discoveries in the area of messenger RNA and stability and translation. He received his PhD in cellular and molecular biology from the University of Wisconsin and was a postdoctoral fellow in the Howard Hughes Medical Institute at the University of Arizona. Prior to moving to Johns Hopkins, Dr. Collar served as a director of the RNA Center at Case Western Reserve University, where he held the Henry Wilson Payne Distinguished Professorship. He studies the very essence of life, translation of the genetic code. His work has led to fundamental shifts in the understanding of gene expression by demonstrating that the genetic code is a major determinant of mRNA fate. He's the co-founder of Tevard Biosciences and Wive RNA Therapeutics. In 2018, Tevard Biosciences was awarded Pfizer's Golden Ticket Award for promising neuroscience startups. His publications have been cited over 6,000 times, and he currently holds numerous patents for RNA-based therapeutic applications. A recorded version of this webinar will be available on SRF's website under webinars on the family menu. And at the end of Dr. Collar's presentation, you will have the opportunity to ask questions. Please write your questions in the Q&A below. And for those of you just joining us, our speaker today is Dr. Jeff Collar. It's now my pleasure to turn things over to him.
Great, thank you, Lauren. You can hear me okay?
Yep.
Okay, great. Let me share my slides. Thank you, Lauren and Mike, for the invitation to talk to you guys today. Let me make sure that we can share and you can see everything. Everything's okay?
It's good.
Good. Okay, great. Again, thank you to the Syngap Research Fund for the opportunity to give this webinar today. I'd like to tell you about some of the work that we've done in the RNA therapeutic space that is being done both in collaboration with Tevard, but then independent work that's being done at my lab at Johns Hopkins, and and that's actually the work that I'll talk about where relates specifically to Syngap. And just as a brief conflict of interest statement, as Lauren said, I'm the co-founder, one of the co-founders of Tevard Biosciences and a member of their SAB. And so, this group needs no introduction to this, but what I have become interested in over the last few years are genetic disorders called haploinsufficiency disorders, where a single mutation within one of the two copies leads to half of the amount of protein that's needed for appropriate wild type function. And the really fascinating aspect about this is that the mutation is not necessarily a gain of function. It's simply that it's reducing the amount of protein, and you need 100% of the protein expressed in order to have a normal individual. And so this seems like it should be a relatively simple fix from a genetic standpoint, where if there's a mutation in one of the two alleles, that all we need to do is raise the amount of protein twofold. And while that seems simple, it's actually very difficult to do. And what I'm going to tell you about are three technologies that my lab has been involved in developing, where what we're going after is, in fact, the mRNA, and in an attempt to either express the mutant copy of the restore expression of the mutant copy of the RNA. And this is specifically through very specific types of mutations called stop codon mutations, or two technologies where what we're doing is, in fact, stabilizing and improving the translation of the wild type copy of the mRNA. And this is the particular technology that I'll spend the most amount of time on today, because it's very powerful for genetic disorders like Syngap, because it's mutation agnostic. And I'll explain what that means in a few minutes. And so if we think about an mRNA, which everybody by this point in the world has heard about, which makes my job a lot easier. mRNAs are, of course, the communicator between genetic information from DNA to protein. They're the intermediate that carries that message, that information, to translate the genetic code into protein. And if we look at features of an mRNA that are important for its stability and its translation, everything that I've shown here in red is a feature that we know contributes to the overall amount of protein that comes from that mRNA and how stable that mRNA is within the human body. And that includes the structure called the cap. It includes what's called the 3' untranslated region, and then codons themselves, which my lab has demonstrated a great deal of evidence about, and stop codons, and then this thing called the poly A tail, and we'll talk about that at the very end of my talk. And so in my lab, what we've tried to do is leverage what we know about mRNA metabolism to improve the overall expression of the mRNA. And we've focused really on these 3 features of the mRNA: the codons, the stop codons, and the poly A tail. And so I'll tell you 3 separate stories about each one of these. And the first part of my talk will be work that I did in at Case Western Reserve University in collaboration, forming TEVARD, and then in collaboration with TEVARD, which has used these unique RNA molecules that are called tRNAs in order to improve the expression of an mRNA. And then at the end of the talk, I'm going to tell you about work that we're doing independent of TEVARD, which is through my lab at Johns Hopkins on poly A tails. And this is really the work that we focused on Syngap. But all of these approaches have the potential to be used on Syngap deficiency, and I'll show why that's true in a few seconds. And so just as a brief introduction to TEVARD Biosciences, this is a biotech company that I was scientific co-founder with Dr. Harvey Lodish of MIT, and Daniel Fisher and Warren Lambert are founders. And both Daniel and Warren have children who have Dravet syndrome. And this group all knows what Dravet syndrome is. And TEVARD, in fact, is Dravet written backwards. And the idea here being that TEVARD's mission is to turn these recalcitrant disorders like Dravet and Rett syndrome and Syngap, to turn them around in some fashion. So this company was started in 2018 in my lab, and then spun out to Cambridge, Mass at Lab Central, and now has grown quite a bit over the last few years, and just entered into a unique partnership with Vertex to look at other disorders, including Duchenne's muscular dystrophy. And so the 2 technologies that we started TEVARD on were the usage of tRNAs to leverage mRNA metabolism. So what is a tRNA? A tRNA is a small RNA in the body, about 75 nucleotides in size, very small. And what tRNAs do is they communicate the genetic code from nucleic acid sequence to protein sequence. So an mRNA carries the information, and that information is read by a ribosome and a tRNA. And that tRNA, there's a tRNA for each of the 61 codons within the genetic code, more or less. There's some redundancy in that, but that's all that's really important to know for today. And the 2 technologies that we have are what we call suppressor tRNAs and enhancer tRNAs. And I'll talk about enhancer tRNAs first. Now, why use tRNAs? We've heard a lot with COVID about mRNA therapeutics. mRNA therapeutics are have been given a lot of interest in the biomedical space as of late, but outside of vaccines, we haven't shown that they have efficacy at this point, but it's likely that they will. But tRNAs are unique in that they can address the root cause of a disease, especially a haploinsufficiency disorder, because they can restore, and in the case of the suppressor, can restore the exact amount of protein that needs to be expressed, or at the very least, They won't go over their target. I'll explain why that's true in a minute. The other thing is that gene therapies through AAV, and everything that we're going to talk about at Tevard is going to be AAV based. Gene therapies delivered by AAV are limited by this 4.7 kb target size. You can't get a gene into an AAV vector that's larger than this. And many human genes are much larger than this 4.7 kb limitation. tRNAs are incredibly small. They're only 75 nucleotides in size. And so that means you could literally put hundreds of tRNAs into a AAV vector in order to reach a therapeutic margin. In addition, one of the things that is the most attractive aspect of tRNA-based therapies, especially a suppressor therapy, is that it's a one-size-fits-all. That one particular formulation could be used to treat multiple rare disorders, such as Dravet, Rett, Syngap, and others could use the exact same therapeutic. And so we shouldn't think about this type of approach as we're going after Dravet syndrome, or we're going after Rett, or we're going after Syngap. We're actually going after most of these things all at once. Using AAV vectors, of course, is a one-time delivery mechanism. And as I mentioned that in the case of suppressors, this actually can restore the normal expression of the gene. And for some disorders like Rett syndrome, it's known that overexpression, going over that normal 100% amount of protein, is in fact just as bad as having the haploinsufficiency. And there are other reasons why tRNA suppressors are in fact useful. So tRNA suppressors and tRNA enhancers can be used to treat a variety of mutations, a variety of indications. As I said, in the case of suppressors, they treat nonsense mutations. And even though disorders like Syngap are the result of spontaneous and random mutation, many of those mutations that occur in these haploinsufficiencies and all genetic disorders are in fact nonsense mutations. It's about 15 to 50% of all genetic diseases are caused by a nonsense mutation. So having a suppressor that can read a nonsense mutation allows you to go after a large variety of genetic diseases. Haploinsufficiencies, of course, there's at least 300. It's predicted up to 1,000 in the human population, and our technology can be used for most of those, we believe. And it really, our sweet spot is somewhere in between here in diseases that are not amenable to gene therapy. And these figures actually just show many of what I just said in the previous slide of why tRNAs are powerful. Okay, so tRNAs are a critical intermediary in the translation of the genetic code. As I mentioned, RNA, mRNA, messenger RNA, is transcribed from your DNA. This mRNA encodes a single gene in the human body. And those nucleotide sequences are then read by the tRNA. The tRNA basically determines, essentially it reads these individual, what are called codons, and parlays that information into protein identity, so that at the end, you get the exact protein that was dictated by the DNA sequence. And so this intermediary is essentially what we're focusing on to improve mRNA biology, improve mRNA expression. And I'll go through why that works right now. So the first approach I'm going to talk about is enhancer tRNAs, and it's really based on work that was conducted in my laboratory from the year 2015 to 2010 to 2015. And for the last 30 years, my lab has been interested in the process of mRNA stability, how fast an mRNA is destroyed by the cell. So when DNA makes mRNA, or when mRNA is made from DNA, while DNA is very stable, the mRNA that is made is unstable. It goes away over time. It only sticks around, for the most part, for a few hours. And the interesting observation that we've known about since the 1970s is that all mRNAs are degraded at a different rate. This is what we call a northern blot, and all it's showing you is an mRNA, and you just need to understand that it disappears. And this mRNA is disappearing very quickly, half-life 3 minutes. This mRNA is very stable, has a half-life of about a half hour. And then this mRNA is somewhere in between with a half-life of about 15 minutes. And if we look at all 26,000 or so mRNAs that we know about in the human body, they all decay at a different rate. And that rate is variable, meaning that it's not quantized like this, where it's short, medium, and long. It's every range in between. And so my lab has been interested in how is that regulated. So why do we care about that? We care about mRNA destruction because if you were to calculate how much of an mRNA is present within the human body, it's function of this equation, which basically is that the rate of synthesis of an mRNA impacts the overall abundance of that mRNA. And that is equal to how much mRNA decay occurs on that transcript. And so an 8-fold change in synthesis of the mRNA will have the exact same effect as an 8-fold rate of destruction of that mRNA in terms of the overall abundance. And now you have to think about this in terms of haploinsufficiency disorders, where we're trying to change abundance. And so if we can modulate mRNA transcription rate, of course, we could change abundance. But another way to do that, which is equally as powerful, is to modulate decay rate. And that's what we're doing. And so what my lab had discovered in 2015 was that the major mechanism for how these transcripts elicit different decay rates is the genetic code itself. That codons can broadly influence mRNA stability everywhere from yeast to humans. And these codons, these individual 61 words within the genetic code, there's actually 64, but 3 of these are stop codons. 61 words are what dictate the identity of a gene, but it's the constitution of these codons within an mRNA that dictates how fast it destroys. And I'll just briefly show you that. And basically how this works is every mRNA has codons within it, and those mRNAs are read by a machinery called the ribosome. And how fast the ribosome reads these codons is what dictates their stability. The ribosome is moving along the transcript, the mRNA's body, deciphering these codons by use of the tRNA. And if that ribosome is moving fast, that mRNA is very stable. But sometimes these codons are difficult to read by the ribosome. And the reason why they're difficult to read is because the tRNA and how much the tRNA is present within the cell is limiting. It's low in concentration. And so the ribosome finds this codon, for example, and has trouble finding the right tRNA. There's a cloud of tRNAs around the ribosome, and it has to sample these in order to find the cognate or the right one. And if it's low in concentration, it's going to hesitate there. And those hesitations are cumulative across the body of the transcript and read by the machinery that destroys the mRNA. And so if the ribosome is hesitating, this machinery binds to the ribosome and then destroys it. And so this is what's called codon optimality. This term was actually coined back in the 1960s as a theoretical possibility. And my lab essentially showed that you can classify codons into either optimal or non-optimal based on pools of tRNAs. And that results in either faster or slower decoding of the genetic code and faster or slower rate of mRNA decay, or opposite actually. And so if we quantitate tRNA levels in the cell, this is actually from another group's, Judith Friedman's lab, and this quantitation of the tRNAs breaks those into optimal codons or non-optimal codons, non-optimal being the ones that hesitate, optimal being the ones where the ribosome moves fast. And if we take how individual codons influence mRNA stability, and we overlay that with tRNA levels, it's a perfect match, such that the codons that stabilize an mRNA are all optimal, meaning that they have large or high concentrations of tRNAs, and the codons that destabilize an mRNA are all non-optimal. Their tRNAs are relatively low in abundance. And the way this looks from a gene by gene basis is it's a ratio of all of these optimal to non-optimal codons that dictates their stability. And this is really kind of the critical experiment for the therapeutic that I'm going to talk about, which is that we can then modulate the stability of an mRNA without changing its polypeptide sequence by simply affecting the optimality of the codons. And here's an experiment where we're gradually replacing codons for synonymous codon pairs that are optimal. And as you can see that in the course of the experiment, we haven't changed the polypeptide sequence. And I'm just showing this experiment twice, that you get a dramatic stabilization of the mRNA just by changing the optimality of those. Now, in the human body, we can't change the optimality of the codon itself by changing the codon, but we can change its perception by altering the tRNA concentration. And that's what we do in TEVR that I'll talk about. And so the idea here is that if we can identify slow codons within a gene like Syngap or SCN1A, which is related to Dravet syndrome, we can find codons that super slow down the ribosome. Can we now express their cognate tRNA to speed up the ribosome? And essentially what that's going to do is stabilize the mRNA. This in fact works. The experiments that I'll show you are really with SCN1A. That's the only test case that we've done. SCN1A has 5 groups of very non-optimal codons, and by expressing their cognate tRNA, we can then improve the expression of SCN1A in a message-specific fashion. And that's just showing over here that these cocktails of tRNAs, it turns out to be 3 unique tRNAs, all shown here in green, will lead to a dramatic increase in SCN1A levels. And this is extraordinarily specific because these 3 tRNAs these 3 codon triplets are really only enriched in SCN1A and not at the same proportion in any other gene in the genome. And so this is a mutation agnostic technology because it's not working on the mutant copy, it's working on the wild type copy of this particular gene. And so it allows us to improve its expression. Now, the other technologies that I'll talk about are much simpler to understand. And the one that has the most promise to get into the clinic the earliest is in fact suppressor tRNAs. And this technology has been advanced by TEVR considerably in the last few years. And just to introduce this, tRNA suppressors are a platform that, as I mentioned, provides a one-size-fits-all cure for a broad range of diseases that are caused by nonsense mutation. And this includes Syngap for patients that have a nonsense mutation. What are nonsense mutations? I've already shown you this is the genetic code that was elucidated in the 60s. And we know that 3 of the codons encode what we call stop codons. This tells the cell, these stop codons tell the cell when to stop making the polypeptide. And there are 3 of them, UAA, UAG, and UGA. And these stop codon mutations often occur in human genetic disorders. And there's particular reasons for that I could always answer another time. But what that ends up doing is that the stop codon causes the polypeptide to prematurely terminate, and therefore a loss of function of that protein in one of the 2 copies in the case of a haploinsufficiency. And so what the game is that we play here is we now take a tRNA, so just to back up, there is no naturally occurring, so tRNAs decode all of these codons in the genetic code, but there is no tRNA for these stop codons in the human body. They are in fact fed by a protein, not a tRNA. And so the game we play in this type of therapeutic approach is to chemically alter a naturally occurring tRNA so that it can now read those 3 stop codons and put back in the right amino acid that was missing from the patient in their protein. And so that's what we call a suppressor tRNA. It turns out if you look at, this is ClinVar data, if you look at all genetic mutations that have been associated with some sort of clinical pathological outcome, most nonsense mutations within the human genome or human population are the result of an arginine codon going from a CGA to a UGA. This would actually be a U. So TGA, is human RNA. And so this stop codon is the most common stop codon that you find in all human disorders. And that's going to be true of Syngap. And these others, of course, are also present at high levels. But if we can develop suppressors for just a handful of these, then we can cover almost 90% of all known stop codon mutations within the human population. And so it does become a one-size-fits-all for a number of genetic abnormalities. And this is really what we would call precision medicine, where we're identifying a therapeutic based on the patient's mutation. Now, just for the aficionados that might be in the audience, they might ask, why do suppressors not read through normal stop codons? And the fact is they don't. There is a mechanism that's poorly understood, but does exist, which discriminates the normal termination codon from a premature stop codon or premature nonsense mutation. And in actuality, if you look at ribosomes and their movement across an mRNA, you can see that the ribosomes will pile up at the normal stop codon. But if you express various suppressor tRNAs, you'll never see that ribosome move beyond that normal stop codon, but it will read through a premature stop codon. And so it becomes an exquisite therapeutic that can be used to target only the diseased allele with minimal opportunity for off-target effects. And because it's only working on the mutant mRNA, and it can only act on the actual amount of mutant mRNA that exists, it will never overexpress the protein. It will only restore the protein at most to wild type levels. And so here's an example of this in practice, where this is a cell line that's expressing SCN1A. This is the normal amount of expression. And this has a mutation. So this is actually haploid. This is a normal amount of expression over here. And we have a mutation in arginine at position 1407. That's taking this CGA and turning it into a UGA, stop codon. And so without a tRNA suppressor, you get no expression of the protein. But if we add back the tRNA suppressor, arginine is restored at that stop codon, and now you get appropriate expression of the protein. And this is comparing Tevart's tRNA suppressor technology to small molecule drugs that have been touted as translational read-through drugs, including these aminoglycosides and adalurin. And you can see that at the levels that we have presented here, and we see very little read-through at all in this case. Not only is it expressing the wild type amount of the protein, that amount of protein is actually, or the cell is now functional. And so SCN1A is a sodium channel gene, and using a patch clamp assay, you can see that when you have a mutant SCN1A protein, you get no opening and closing of that sodium channel. But when you restore the arginine at that stop codon mutation, you now get the appropriate wild type opening and closing of the sodium channel. And so in fact, it restores protein activity and protein abundance and activity. One of the beauties of suppressors is that, as I said, you can use this for all types of disorders as long as it's the same stop codon. And so here's an example using SCN2A. And these are various mutations, all arginine stop mutations, arginine CGA going to UGA at different positions within that gene, and expressing the exact same suppressor tRNA. And that's important. Every one of those is being restored to a wild type function. And so not only can you do this within a cohort of patients that have the same mutation, but at different positions, you could also use the same suppressor across indications. And so that was Dravet syndrome. And now this is Rett syndrome that's linked to the MECP2 gene. And in the case of MECP2, this assay is essentially looking at expression of MECP2, which is a nuclear localized protein. And in the absence of a suppressor, you get very little expression of MECP2. But when you express the exact same suppressor that I just showed in the SCN1A and the SCN2A, we're getting the same restoration of MECP2 function. And so it's a drug that can be used across indications, including in Syngap patients who would have a UGA mutation. And just to point out, Tevard is working on all 3 stop codon mutations, and we'll have suppressors soon for most of those in the next year. Okay, so just to wrap up, this is the work that has been done by Tevard in collaboration with my lab back at Case Western. And it's tRNA-based therapeutics that have the promise to ameliorate many haploinsufficiencies and other genetic disorders based either on improving the wild type mRNA's expression or helping to restore the expression of alleles that have nonsense mutations. And so now I'd like to move away from the work that we've done with Tevard and talk about new technology that my lab has developed in the last 6 months at Johns Hopkins. And this is really work that we've done in collaboration with the Syngap Research Fund through the funding that they have so generously provided us. And this is what we call an mRNA booster therapeutic. And so not only was this supported by the SRF, also I'd like to give thanks to Carl Hall and Mrs. Nancy Hall for a generous charitable donation, as well as the Bashadi Translational Fund here at Johns Hopkins University. So what are mRNA boosters? So in the last few years, I've thought a lot about genetic therapies linked to AAV vectors. And there's a lot of promise of AAV-based approaches. It's a one and done. But it may not be always the right choice, depending on the indication. And I've wanted to develop novel therapeutics that could be delivered not by AAV, but by a nanoparticle or an LNP, a therapeutic that could be transient, a therapeutic that could be used to improve expression of an mRNA for a brief period of time, or be manipulated to express an mRNA over a long period of time, but that we had much more control over the chemistry of the RNA. And so returning back to the identity of what an mRNA is, as I mentioned, mRNAs have caps and they have codons and they have poly A tails. And what we focused on for this technology is in fact the poly A tail. What is a poly A tail? Well, every human mRNA, with the exception of a handful, we don't need to talk about what those are, almost every mRNA has a poly A tail. And the function of that poly A tail is to drive the translation of the mRNA. The poly A tail is variable in size. It usually is about anywhere between 50 to 300 nucleotides in length of just poly A. And as long as that mRNA has a poly A tail, it will bind to ribosomes, and those ribosomes will translate making polypeptide. And you'll get protein expressed from that fully functional mRNA. But as soon as that mRNA loses that poly A tail, so that it is now what we call deadenylated, no protein is made anymore. And that mRNA has one of two fates. Either it's translationally inactive and stored, or most of the time it's destroyed. And the function of the poly A tail is in fact to act as a slow burning fuse that dictates how long an mRNA is going to express a protein. There are enzymes that are constantly degrading the poly A tail as soon as the mRNA reaches the cytoplasm. These are called deadenylases or poly A shortening enzymes. And this is a clock. The rate at this removal is a clock. tells the cell how much protein should I make. And as long as that tail is there, the mRNA makes the protein. And as soon as the tail is gone, it stops making protein. And so the goal here is to trick the cell into thinking that this poly A tail remains on an mRNA and continues to translate. And so that's what we call an mRNA booster. And what we have done is we've taken synthetic RNAs where we have a short guide sequence. This is about anywhere between 30 and 50 nucleotides in size. And it specifically binds to the 3' untranslated region of an mRNA. And when it binds, what it's doing is supplying a poly A tail mimetic. And we can modify this transcript to maintain its overall stability within the cell and improve the gene expression of that mRNA. And so what this booster RNA transcript does is it fools the cell into thinking that this mRNA still has a poly A tail and should still make protein longer than what it normally would from its normal endogenous poly A tail. And the benefit of this is that this is really small. This is only about between 70 to 100 nucleotides in size. It's very analogous to what you might think of as an antisense oligonucleotide, but it's not the same. This is in fact an RNA that's improving translation of an mRNA out in the cytoplasm. So this technology works. Here's one of our initial experiments that we did in vitro, and we're doing this again on the MECP2 gene, MECP2 linked to Rett syndrome. And this was just a test of our technology and various ways in which we organize the poly A tail relative to the guide and different modifications we put on it. And essentially what you can see is that this particular booster is the winner, where if we have a modification present on this 5' end, and we have the poly A tail mimetic going this way, followed by the guide, we get a dramatic increase in the expression of that protein within this cell line. And that's like a 10-fold increase, very dramatic. The same exact booster, now we don't see the sort of dramatic effect in vivo, which is important. That same exact booster can be injected into a mouse. And in this case, what we've done is we've injected into the tail of a mouse. And we have 3 mice that we've injected controls, and 4 mice, we inject 1 booster, and 4 more mice, another booster. And if we look at the amount of mRNA expression that we see with this booster 2 that I highlighted in the previous slide, we get an increase in the mRNA stability. And if we look at the actual protein that comes from MECP2, or from that mRNA, the MECP2 mRNA in the liver, this is specifically in the liver, then you can see an increase in protein amount. You can see that this is just darker. And so in a mouse, we can improve the expression of MECP2 expression in the liver. This technology is portable across indications. And what I mean by that is, if I go back to my slide here, all I have to do is change this guide sequence to a new gene. And so in the previous example I showed you, this would be complementary to the MECP2 gene, but I can change this to any gene that I want, any mRNA sequence that I want, and redirect the booster to that transcript, that mRNA. And so here's 2 additional cases. These are genes also linked to haploinsufficiency. This is the gene for beta-catenin, CTNNB1. And this is a gene linked to a very ultra-rare haploinsufficiency called pure alpha. And in both cases, we can develop boosters that will enhance the expression of those specific genes in a specific manner. So what about Syngap? So we've done a lot of work on Syngap with this particular technology. I don't need to introduce Syngap deficiency to this crowd, but Syngap is like many of these haploinsufficiencies, that it's a debilitating monogenetic encephalopathy. It does have some similarities to other disorders that I've talked about. And if without the right expression of Syngap, we see an increase in the excitability of synapses, which makes it difficult for neurons to communicate. And this is what often leads to many of the issues that we see in Syngap patients. The phenotypes obviously everyone knows this, phenotypes of Syngap deficiency are shown here. This includes moderate to severe intellectual disability, um, tremors, and epilepsy, as well as autism-like behavioral issues. And one of the things that is really difficult with many of these disorders like Syngap and Dravet and Rett syndrome, et cetera, is that they tend to result from sporadic germline mutations. They're not necessarily inherited. They can be, but they're not necessarily inherited. It's a sporadic mutation. And this causes a problem when you try to develop gene therapies, because there are patients that have all sorts of mutations. They have stop codon mutations, they have missense mutations, frameshift mutations, splice site changes. And so how do you develop a gene therapy to deal with this? And the trick is to really go after the wild type RNA or the wild type gene. And that's essentially what the booster technology allows us to do. In addition, like many genetics, like many genes in the human genome, there's one gene, but it actually produces multiple isoforms. And so there are 19 exons in the Syngap gene, and those are stitched together to make the full-length isoform of Syngap1. But then there are other minor species that also exist that are the result of alternative splicing. But one of the most important things about, so what this means, and there's some evidence for this in the Syngap community, is that the expression of all four of these isoforms is in fact important for wild type function. And so how do we improve the expression of all four? And what's important here from our standpoint is that they all share the exact same 3' untranslated region. And this is the region that we target with our mRNA booster. And so we've developed a mRNA booster that specifically recognizes the Syngap mRNA, and that's shown here, where we have this 2, SB1 and SB2, Syngap booster 1, Syngap booster 2. And in the case of mRNA levels, this is actually done in vitro. These are in these SHSY5 cells, which are a neuronal line. We can get a dramatic improvement in the mRNA's expression in the cell culture system. And so now what we're doing with this technology is we have to go through a lot of pieces, a lot of things to do to make sure that it's going to work and that it's going to work in a clinical situation. So we use in vitro systems, like these cell systems that I just showed you. We use patient-derived iPS cells and we've gotten some of these from the SRF. And thank you to the donors who have given us those. We will take those iPS cells and turn them into neurons and then test our technology. And once we develop a technology, we have to de-risk it, do lead candidate optimization, and then what is important is we have to test this in mouse models. And so one of the aspects that we've gone through in testing and doing lead candidate optimization is to find boosters and that give us the biggest amount of mRNA expression in the Syngap1 mRNA. And so in this case, we've done a limited scanning of the 3' UTR, where we have 5 boosters and some controls. And you can see that this particular booster here is giving us the biggest increase in overall expression. Now we've actually gone ahead and taken one of these. We actually started with SB1. We haven't gone on to these next guys yet, but we started with SB1 and developed that further in mouse model systems. So SB1 is nice because, well, the Syngap mRNA is nice because it is 82% identical in the 3' UTR between human and mice. And this becomes really important for us because that means that we can do experiments in the mouse and take that information and translate it right back to the human. So this particular booster, SB1, binds with perfect complementarity to this region in the human 3' UTR of Syngap1, and that's identical to the same region in the mouse 3' UTR. And so that allows us to test this clinical candidate in a mouse. And so what we've done in this first set of experiments, there's a lot of things we can do with this technology. We can make LNPs and nanoparticles and try different routes of administration. But in our first set of experiments, we wanted to do a direct deep brain perfusion of our booster into the hippocampus. And this has been done in collaboration with our colleagues here at Hopkins, Yicheng Len and Heshuan Mao over in the Institute for Nanobiotechnology on the Homewood campus at Hopkins. And Din Chin's experiments, what he has is a really unique system where he can, using artificial intelligence, and I don't pretend to understand how this works, but he can develop a device which has a stiff tube and a super elastic tube encoded inside that tube. And then what you do is you insert that stiff tube into the brain, a single point of penetration, and then insert this super elastic tube. And it's here where the artificial intelligence has plotted out the path it needs to go in order to move through the hippocampus. And so this tube has been engineered in a way that as it's pushed in, it moves in this pattern. And this has been shown as a curve for simplicity. But then what happens is that when you retract that tube, it leaves behind this super elastic nanotube that then can be perfused with small molecules, or in our case, a small RNA that has been encapsulated in a neuronal specific LNP. And the neuronal specific LNP was developed by Heshuan Mao over at the Homewood campus. And so we've done this deep brain perfusion experiment with our booster, and you can see sort of two results in this figure. We injected into the hippocampus and then dissected the hippocampus, the cerebral cortex, midbrain, and the cerebellum from these mice. And this is the mRNA quantification, but really what's important is to look at the protein quantification. And I'm only showing a Western for the full-length Syngap 1 protein. And camera's a little bit in the way, but what you can see here is in the hippocampus, you get a nice increase in the expression of Syngap 1 that telegraphs that the booster is telegraphing into the cerebral cortex and the midbrain. But by the time it's just the cerebellum's way too far away and we're not having much of an effect there. So there's actually not much Syngap expressed in the cerebellum, at least in our experiments. And so this gives us a lot of hope that this technology will work and we can further develop it and modify the RNA to increase not only its efficacy, but its durability. We need to make sure that this can last a long time in a mouse. Will the mouse express Syngap at this level for days, weeks, months? What? We haven't gotten to that point. But those are our next steps, is to improve the overall durability of the small molecule. And then also to de-risk it in terms of making sure that it doesn't have off-target effects. We'll do both the RNA-seq on this as well as proteomics to ensure that we're only having this effect on Syngap expression. And then from there, we'll take it into model systems. These experiments were all done in wild-type mice, perfectly happy mice. We'll take it into mouse models where they have a Syngap deficiency. We didn't want to do that right now because those mice are very precious and they're delicate, and we really want to do a lot of our tinkering with the booster in the best case scenario, which is really a wild type mouse. Okay, so I have 10 minutes left for questions. Let me just summarize by saying that this new technology that my lab has developed, we call an mRNA booster. It is a novel gene therapy that enhances gene expression. It really is a first-in-class technology. We're not aware of any other technology that does this. And it leverages the power of RNA therapeutics in order to do what it does. We believe it'll be disease modifying because it restores the haploinsufficiency. It also is mutation agnostic as it works on wild type mRNAs. And it really is a platform technology. We've chosen to develop this for Syngap, but this could be used in theory for any one of these haploinsufficiencies that I've talked about today, or the 300 other that I haven't. And there are also other applications for this technology that go beyond the treatment of haploinsufficiencies. So, okay, with that, I'll just thank the people who've done this work. This was all done at Johns Hopkins. The booster technology was all developed at Johns Hopkins. Everybody here is past or present member of my lab. The present members are highlighted in asterisks. And these are the people who give us the money to do what we do, and especially the Syngap Research Fund, without whom we never would have gone down this road and tried to develop technologies that really can help this particular disorder. So thanks to you for all of your efforts, your precious donations, and the time and reagents that given to us. So thank you.
Awesome. Can you guys see me? Hear me?
I gotta stop share. Here we are.
Okay. Thank you so much, Professor Collar. That was incredible. And I did not prompt or pay Professor Collar to talk about the value of reagents or the importance of us being able to support work like this when it comes to our attention, but they're valuable, and see previous discussion on making cell lines. So people have jumped in on the Q&A, and I imagine the parents are a little overwhelmed at this point if they're seeing this for the first time, but I want to encourage the parents to ask questions. As we have seen today, Professor Collar is adept at making the incredibly complicated accessible, and I urge you not to hold back. But let's go through the Q&A while people are collecting their thoughts. And before we do that, I just want to, because you covered 3 technologies, I think some people might walk away from this and say, okay, only if my patient has a nonsense, which is an R stop or a Q stop, is this applicable to me? And can we just clarify at the highest level the different things in the poly-A, which ones work, which ones are mutation specific and which ones are mutation agnostic?
The only one that's, you can still hear me, right? The only one
the
only one that's mutation specific is the nonsense suppressor.
Right.
That's only for patients that have stop codon mutations. But in some indications, that represents a lot of patients.
Yes.
And one of the beauties of that, of course, is that once it's approved and been shown to work in Dravet, the same exact formulation could be moved to other indications. And so you can do all the de-risking of the technology, the talks, everything on one indication, and then move it through everything else, Rett, Syngap, go down the list of neuronal encephalopathies that could benefit from that. And so by having one therapeutic, maybe it doesn't help as many patients within Syngap, but collectively, it's helping lots of kids. That's the beauty of that technology.
Right, but the poly-A would be mutation agnostic. So anyone with a frame shift or.
Right. That's exactly right. And the differences between those technologies, the enhancer and the booster, the enhancer is a AAV-based approach, and the booster is LNP nanoparticle approach. And so what it really does is gives you multiple shots on goal of what's going to work, right? If in my career, what I've decided to do is not stop with one, I'm not one and done, that I'm going to go after a particular technology. And I think the model that works better for researchers like me, develop a technology, then give it to biopharma to de-risk and do all their development of. That's what Tevard's doing with the enhancers and suppressors. The booster technology that, we'll talk about that at another day, where that will go. But that's really the point is, and we're not the only game in town. Stoke has ASOs and other groups are developing ASOs. And hopefully, one of these things works. And, it's, the more opportunities we have, the better chance we have to get success. I mean, look, making a gene therapy is not easy. It's tough.
Yeah.
It's tough.
Yeah.
Lots of mice have been cured.
Yeah, no. And another time we should talk about Zemplenny. There was another SRF grantee where we've talked about delivery. And as I think about sticking your tech in his delivery, it'd be pretty exciting. But let's stay on topic. So Ashish, TP53 you've really woken him up and I have no idea who this is, but he has a lot of questions. And then, but before we go to him, there's the anonymous questions about missense. Can you briefly address why or why not this might work for missense mutations? Same questioner, has to do with the, yeah.
I'm not, I don't know exactly what technology they're talking about, but I'll just answer that. So the suppressor will not work for missense mutations. That it can't. Missense mutation is a mutation that made the wrong amino acid, turned it into something else, right? But the enhancer and the mRNA booster would work for patients with missense mutation, because again, it's helping improve the wild type copy. As long as that missense mutation is not leading to what we call a dominant negative, which for a lot of these, we don't know all the time whether that's true, but we suspect that most of the time these missense mutations are simply a loss of function, meaning that the protein no longer works. But to be honest, I've talked to some very smart people recently, and they even think that with some of the missense mutations, it's sort of a partially functional protein, and maybe improving its expression would give us a little bit of a therapeutic window. So, I think, and that's, in fact, we're going to test that with our RNA booster on that very idea, whether improving some of these missense mutations will give us a therapeutic margin. So hopefully that answered your question.
Well, there's people in the audience, so I'm curious if you just triggered a question. But so Ashish's questions, Ashish has a lot of questions, but I like his last question best because he's asking you, which of your children is prettiest? So picking one of the three, booster, enhancer, suppressor, who's going to make it to clinical trials first? It's a hard question.
Well, I have 3 daughters and they're all beautiful, and I have 1 boy and he's beautiful too. But I mean, my pet project right now, I don't know. I think they're all wonderful technologies. Who's going to reach the clinical stage first? That's actually easier to say because it's going to be the suppressors. That's already advanced. I mean, we started Tevard in late 2017. It's been, what is that? 6 years of development. That will get the clinic way before anything. The booster technology is still within my lab. It's not even ready for, there's a lot we have to do before biopharma picks that up. Suppressors are, most of this work on enhancers and suppressors, since it's not done by my lab anymore. That was developed back in 2018 in my lab, and it's moved on to Tevard. They do their stuff with it. So.
And is the poly-A stuff delivery agnostic, mode of delivery agnostic? Like, could you put it in multiple?
Theoretically, you could put it in an AAV. In theory, we have actually shown, I didn't show these experiments here, but we've packaged it into a nanoparticle that our colleague made. We packaged it into an LNP that our colleagues made, and it works in both cases. And we've shown that it works in the liver. We've shown it now works in the brain, 2 different genes. We could put it in AAV. I see no reason why you couldn't, but we haven't done that yet.
Got it. Do you want to go through Ashish's questions, or do you want me to read them? It's a lot.
I can answer some of them. So let me start from the back. How do you choose among the various booster guides? Do we need to perform wet lab experiments to find the most optimal guide? Yes, you always have to perform wet lab experiments to make sure. Can use computational approaches and machine-based learning to play a role to arrive at optimal boost guides faster? Potentially. But I'm an old school molecular biologist. I don't think you have to do the experiments because for as smart as we think we are and as smart as we think AI is, biology is smarter. So you always have to do wet lab. And this is very important because you never know when you're going to get off target and tox associated with a particular small RNA or molecule. So wet lab experiments need to be done. Okay, so that. Okay. Is Tevard your lab trying to apply the tRNA suppressor enhancer approaches to other rare diseases? Yes, it is. They have to talk about their candidate They have to talk about what they've announced already. I mean, what I can tell you is that Dravet is their number one indication of choice, and Duchenne's muscular dystrophy now with our collaboration with Vertex. They are working on other rare diseases, but those programs haven't been announced. Okay. How would you rank enhancer tRNA suppressor technology in terms of the following factors: time to develop a drug, cost to develop a therapeutic drug, efficacy, adverse risk, and side effects? I think I answered that question in some sense. I mean, the suppressor technology is the one that is going to result in the drug fastest. Okay. And then the first question: would it be fair to summarize as a layman the enhancer technology as increasing protein levels by modulating the functional wild-type copy of the gene in question? Yes, that's exactly what it does. It's essentially improving the stability of the mRNA. Because the mRNA stability goes up, protein level goes up.
Super. Everyone else has just been wowed into submission. I'm fascinated. I'm very grateful, Professor Collar, for doing this. I think this is a webinar people will go back to. I thought your isoform slide was really accessible too. I don't think people always get that. And so I guess on the isoform slide, Mike, given that this is so specific, would the different isoforms still be expressed? I mean, would it?
Yeah, I don't have the data to show you because it's red hot off the presses. And you know, I always want to see things a couple times before but
Yeah.
When we've looked at the other isoforms, we're seeing that they go up as well. And it's just what Syngap is, you know, as you know, some of those isoforms are really close together in size. And so it's difficult to resolve. Like when I show that band on the gel, that's probably actually the two isoforms, the two biggest isoforms. I'm not using the antibodies that I know that there are researchers that have developed antibodies that are for the specific isoforms. I don't have access to those antibodies. But it looks to me like at least the visible isoforms that I can see, which I think at least two are both going up.
Yeah, so I wasn't even being that specific. I just framed the question poorly. What I was trying to say is, given that this is affecting the way the proteins are made, there are other therapies out there that are just delivering alpha-1, right? One specific isoform. And then people are saying, oh no, what if we leave out the other isoforms? Whereas with these approaches, am I safe to assume that we don't have to worry about that because the body will still express the isoforms that are needed?
That's right.
Okay.
That's the theory. That's the theory. Because it's binding to the 3' UTR and the 3' UTR is shared between the 4, they all should go up. And so, yes, it should be more amenable to disorders and genes that have multiple isoforms but share common 3' UTRs.
Superb. Thank you very much. This is your last chance, people. No further questions. We're grateful for your time, Professor Collar.
I'm happy to be here, and you know, people can always ask me questions by email. You just Google my name, you'll find my email address.
Great. Thank you so much for your time.
Thank you, Mike.